Okay, should we start now?
Yes, please do. Well,
okay,
please do. Thank you.
So welcome everybody. Today talk is today presenter is Ana Rossi. She's a senior research associate in the Department of Pharmacology at the University of Cambridge and a fellow of Queen's College. She completed her undergraduate studies in Argentina and PhD in Cambridge. She has contributed to the development of new Ip3R ligands to understand the mechanisms of Ip3R activation and also to develop new methods to quantify this activation. Her current interest now is the interaction between hypoxia, Ip3R2, and the invasion of glioblastoma cells. So with you, Ana Rossi now. Thank you.
Thanks, Raul. It's a bit odd, I hope, because I can only see my screen, so I can't see who's there, but thanks those who are listening, and thanks to Martin and Katya for organizing this webinars. So I'll start now, and as Raul said, I'm going to talk to you about my work on the role of type 2 Ip3 receptors and hypoxia and glioma cell invasion. So gliomas are the most common primary brain tumor, and within them, glioblastoma multiforme, or GBM, that is the grade 4, according to the World Health Organization, has a worse prognosis. And despite advances in multimodal therapies, that is surgery, chemo, and radiotherapy, these tumors remain fatal in most patients with a median survival time of 14 months. And little has changed over the last 2 decades. So these tumors are very aggressive, and partly because they are very invasive. So although they rarely metastasize outside the brain, they invade surrounding brain parenchyma, and they almost always relapse even after debulking surgery. One of the characteristics of GBM is seen here in this MRI scan are these hypertense regions surrounding the tumor mass. And these regions are indicative of diffusive invasion. As most solid tumors, glioblastoma or GBM are highly hypoxic. And one of the features that distinguishes GBM from lower grade tumors are found within this ring. And these are these microstructures called pseudopalisades, which are necrotic cores surrounded by radially oriented cells. These structures are believed to form around tumor blood vessels, and tumor growth around these vessels result in vascular injury and thrombosis, and the formation of a necrotic hypoxic cores, and the pseudopalisade cells seem to be migrating away from this hypoxic core. These cells, which express high levels of hypoxia inducible factor, a transcription factor that mediates or orchestrates responses to hypoxia, show decreased levels of proliferation and apoptosis, and they are resistant to conventional therapies. So in order to invade, glioblastoma cells start in a hypoxic microenvironment. They need to loosen or lose cell-to-cell adhesion, then degrade extracellular matrix and migrate. And GBM cells can migrate as either individual cells or loosely connected networks of cells. As I mentioned earlier, invasion is a major clinical problem, but there's no treatment to target these invasive cells. Another problem is a huge inter-patient heterogeneity in terms of invasion, yet all patients receive the same treatment. And this is not least because we do not know the mechanisms that these cells which these cells acquire these invasive properties. As I said, invasive cells have decreased rates of proliferation, apoptosis, they're resistant to treatment, and we don't really know which signaling cascades. Probably several signaling cascades are involved in orchestrating these events. When we started this project, there were several reports suggesting a role for calcium signals in all these processes that I mentioned: cell-to-cell adhesion, cell migration. So we started by looking at the effect of calcium in this. I'm just going to have a brief introduction about inositol 1,4,5 phosphate receptors, or Ip3 receptors. In non-excitable cells, calcium signals rely on Ip3 receptors, which are intracellular calcium channels, which are expressed mainly but not exclusively in the endoplasmic reticulum of all cells, which is a major calcium store. So different agonists acting via GPCRs, or receptor tyrosine kinase, lead to activation of different isoforms of phospholipase C, leading to hydrolysis of Ip2 in the plasma membrane and generation of Ip3. Ip3 together with calcium then bind and activate Ip3 receptors, and this results in calcium release from the ER. Calcium, as we all know, is responsible for regulating a variety of processes from proliferation, cell-to-cell adhesion, invasion, gene expression, and so on. Something that is important for the rest of my talk is to mention that in mammal cells, there are 3 isoforms of Ip3 receptors, Ip3 receptor 1 to 3. They can heteromerize, and although they share high homology and they're very similar in many ways, they both need calcium and Ip3 for activation. They're also different in that they have different affinities for Ip3. The patterns of expression and subcellular localization is different, and although we don't understand it fully, they might play different roles in cellular physiology. So we started to look at the roles of Ip3 receptors. As I said, there were some reports suggesting a role for Ip3 receptors in migration. So we started to look at Ip3 receptors in glioblastoma cells. I'm going to come back to these later, but just to say that we chose initially, and most of the data I'm going to show you is done in these cell lines, 2 cell lines, C6, which is derived from rat, and U87M G cells, which are derived from human tumors. These are very well established and characterized models of GBM, but they're not perfect. So each of them has its limitations. So I'll show you a little bit at the end how we're trying to validate this with patient-derived tumors, glioblastoma stem cells. So we started by looking at whether these cells express Ip3 receptors, and we did quantitative Western blots to determine the relative expression levels of these cells. So for the C6, we used DT40 cells expressing a single subtype of the Ip3 receptors and antibodies that recognize either one subtype or the 3 subtypes, a common antibody. And then by getting a ratio between the signal from the single subtype versus the common in cells that express one subtype, which could determine the relative expression levels. For the human cells, we use hex cells expressing a single subtype, the 3 single subtypes of Ip3 receptors. And what we determined was that in both cases, the type 2 Ip3 receptor was the minor subtype. The expression level was less than 2% of the total Ip3 receptors in these cells. So the next step was to look at the role of Ip3 receptors in cell migration, and we started by looking at collective cell migration in assays which we call wound healing assays. So here in these assays, what we do is we grow cells at either side of the septum in this insert. Once the cells grow to a monolayer, we take the insert and there's a cell-free gap, and then we monitor how cells move to close that gap or close the wound in time lapse, and we can quantify that in an automated way. So interestingly, when we use siRNA to knock down the 3 at the time, one Ip3 receptor subtype at the time, and I'm not going to show you the data, but we corroborated that each siRNA was specific and selective for each subtype, and there were no compensatory mechanisms. So when we use the siRNA for the R1, we decrease the levels of the R1 only and not the other two, and the other two, the expression levels of the other two were the same as the control cells. Anyway, so this was quite interesting because, as I mentioned earlier, type 2 Ip3 receptors are the minor isoform, but only siRNA against type 2 Ip3 receptors had any effect on wound healing, and this is the quantification. So we use 3 siRNAs against the type 2. Here I show you the efficiency of siRNA knockdown. As you can see, 2 siRNAs are knocked down about 50 and 60% of the type 2, reduced the migration rate by nearly 50%, whereas another one that was very ineffective in the knockdown didn't have any effect. And as you can see here, knocking down most of the R3 or the R1 didn't have any effect. So that was quite interesting. Now, the wound healing is the result of cell proliferation, cell-to-cell adhesion, and individual cell migration. And I'm going to cut a long story short by telling you that we try the siRNAs against different subtypes in proliferation assays and single-cell migration. We didn't find any difference. Now, for the efficiency of migration as a monolayer, cells need to be able to maintain dynamic cell-to-cell contact. So the example here, cell contacts are okay, the leader cells drag the followers, and they drag the monolayer forward. Whereas when there's defect, there are defects in cell-to-cell adhesion, migration can be abnormal. So the first clue came from looking back at the videos. I'll try to play them all together, in which if you look at the siRNA control and the siRNA against type 1 and type 3, it seems that the monolayers do move as coherent sheets. Whereas looking at the videos of the siRNA against type 2, we could see that cells were less able to maintain cell-to-cell contact, and many cells could actually escape from the monolayer, and that's what caused the effect in wound healing. Now, we couldn't really quantify these, although we tried. So then we thought, okay, well, if there's a defect in cell-to-cell adhesion, let's measure cell-to-cell adhesion directly. So we started by optimizing an assay, which is a well-established assay to measure cell-to-cell contact, which is called the aggregation assay, in which we incubate a cell suspension in agarose-coated wells to prevent cell to extracellular matrix adhesion, and then we rotate them for about 24 hours, and then we measure the size of the aggregate. So cells that adhere well, they produce these tight aggregates, and we can quantify that. We sort of validated by using EGTA to chelate calcium, which is needed for cell-to-cell adhesion. And then we also put calcium back just to make sure that this was not due to cell death, but a defect in cell-to-cell adhesion. And that's a quantification. So we next try our siRNAs in both cell lines. And again, only the siRNA against type 2 led to looser and bigger aggregates, as you can see here in the quantification. So at the moment, I'm optimizing like some more acute cell-to-cell adhesion to measure this in an acute way. And we are carrying out mass spec to identify the adhesion molecules involved. This was too preliminary to show you today, but I can tell you a little bit about it at the end. Now, so we have cells that knocking down the type 2 Ip3 receptor leads to less to cell-to-cell adhesion, cells that can escape a monolayer. What does that mean for invasion? So we started by using these 3D spheroids, which are nice in vitro, of course in vitro, but nice in vitro models of GBM. And we grow this in low attachment, round bottom wells, so suspension of cells which form a little spheroid. Then we can add Matrigel, for example, to sort of resemble the brain ECM. and then we can quantify in a semi-automatic way invasion from these spheroids. Something that is important for the second part of my talk is that these spheroids are nice in recapitulating GBM because due to lack of diffusion, they intrinsically generate these necrotic cores surrounded by hypoxic areas where the oxygen concentration goes down, going from the outside to the inner part. And this is quite a nice feature of these spheroids, and I'll come back to this a bit later. Now, when we use these assays to check the effects of the siRNA against the subtypes, we found quite a striking phenotype only with the siRNA against type 2, suggesting that knocking down the type 2 Ip3 receptors resulted in more invasion. So sort of nicely tied up together all the data that I show you. So far, I show you that if we knock down the type 2 Ip3 receptors with siRNA, we have decreased cell-to-cell adhesion, and that leads to increased invasion. Now, the role of hypoxia in driving tumor progression and aggressiveness is quite well established, particularly through HIVs and driving all these processes, particularly invasion, which is the one I'm interested. Now, most responses to hypoxia, not all, but most are mediated by hypoxia-inducible factors. We know of 3 types, but the type 1 is the most widely expressed and obviously the one we know most. So HIF are heterodimers of an alpha and a beta subunit. Under normoxic conditions, so normal oxygen levels, both subunits are constitutively expressed, but the alpha subunit is in an oxygen-dependent manner hydroxylated by these prolyl hydroxylases that need oxygen, iron, and 2O-glutarate, and this triggers degradation via the proteasome. Now, under hypoxic conditions or low oxygen levels, obviously these prolyl hydroxylases are inactive, therefore the alpha subunit gets stabilized, binds the beta subunit, and together they translocate into the nucleus to regulate the expression of genes that are responsive, hypoxia-responsive elements. And just as a reminder that I show you that these typical structures found in GBM are thought to occur through hypoxia in this the intrinsic hypoxia of these regions. Now, how do we generate hypoxia in the lab? So we can either obviously stabilize HIF by incubating our cells in low oxygen concentrations, or we can also use what we call hypoxia mimicking agents, that chemicals that either chelate iron, like deferoxamine, or DMOG, that competes with the O2 glutarate. So in that case, we can stabilize HIF1 alpha and then see what the responses to hypoxia are, and we can actually measure HIF1 alpha activation, stabilization by either immunocytochemistry here and Western blotting. I have to say that this is by no means trivial because HIF1 alpha is a very unstable protein, so not so easy to get nice blots all the time, but there you are. So the first thing was just to corroborate this idea that our spheroids have a hypoxic core, and I'm showing you here some images of the spheroids, 48-hour spheroids, showing that they do develop hypoxic cores. This is a red hypoxia reagent, which is actually an oxygen sensor, which fluorescence intensity increases when the level of oxygens go down. And so we we also can downregulate HIF1 alpha by using HIF inhibitors, like for example KC7F2. When we incubated these cells in our spheroid assays with the hypoxia, in the HIF inhibitor, we could see that invasion was reduced, so corroborating the role of this intrinsic hypoxia in driving invasion. Now, when we treated our cells either under low oxygen concentrations or with hypoxia mimicking agents, we noticed that in all the assays we try, these two phenocopy phenocopy the effects of knocking down type 2 Ip3 receptors, and we can see here by a decrease in the rate of wound healing, and also we can see these cells escaping from the monolayer, sort of disturbed monolayer with the treatments that resemble what we saw with the siRNA against the type 2 Ip3 receptor. And also in our aggregation assays, we could see that hypoxia and the hypoxia mimicking agents led to larger and less compact aggregates, and that's a quantification, so suggesting that hypoxia and hypoxia mimicking agents phenocopy the effects of knocking down the type 2 Ip3 receptors. We also looked at the effect of these treatments in the levels, expression levels by Western blot of the 3 subtypes, and again we saw that either treatment reduced selectively the levels of the type 2 Ip3 receptor, and that's the quantification. So although we haven't yet shown the link between hypoxia and the type 2 Ip3 receptor, a working hypothesis is hypoxia by down-regulating the type 2 Ip3 receptors, which is, as I said, the minor isoform, might lead to decreased cell-to-cell adhesion and increased invasion. This is something obviously we need to check. Now, as I mentioned at the beginning of my talk, most of these studies were done in established cell lines, C6 and U87 cells, but each of them has limitations. Now, one of the most accepted models of GBM at present are GNS cells. These are glioma neural stem cells, which are extracted from patients, and they are maintained in serum-free, medium, and low concentrations of growth factors. Now, one of the problems of GBM, another problem, is the intra-tumor heterogeneity. And this is believed to arise and be maintained by these glioma stem cells. So we started validating all the results we got with the established cell lines in these GNS cells. We've got 4 at the moment. I'm going to show you data from one of them. This is more preliminary. And again, we corroborated that the type 2 Ip3 receptor is the lowest, the minor subtype, with less than 2%, like like the same feature of the other 2 cell lines. And again, when we knock down the type 2 Ip3 receptors by shRNA in aggregation assays, we see that these cells are less able to form aggregates. And similarly, when we treat the cells with hypoxia, these aggregates are, cells are less able to form aggregates. This suggests that either hypoxia or knocking down the type 2 Ip3 receptor leads to less to cell adhesion in these GNS cells. And this is very preliminary, since we're trying to knock out type 2 Ip3 receptors with using CRISPR-Cas. We've only managed so far to have a partial knockdown, and we're trying to sort of obtain monoclonal cell lines from that partial knockdown. But this is a preliminary data, which looks very encouraging, in that we have again, a phenotype whereby knocking down type 2 Ip3 receptors in these cells leads to increased invasion in 3D spheroids. And the other piece of preliminary data I wanted to share with you today, this is a more recent collaboration we started with Harry Bolstrode in Addenbrooke's Hospital, who is a neurosurgeon, and he's been working in developing this brain organoid. So as I mentioned, the 3D spheroids are very nice in vitro models of the disease, but obviously we know that the microenvironment plays a crucial role in tumor progression. Now, these brain organoids, which are formed by growing human stem cells under the right conditions for neural development, they sort of self-organize in what people call mini brains. And as you can see here, they show markers of neural stem cells like SOX2. And what is very nice, which Harry has been working on, is that if you incubate GNS cells labeled in green here with these spheroids, these organoids, they recapitulate the disease. So they form infiltrative tumors that recapitulate the human disease. So they're quite nice models to work. So the plan here is to get our CRISPR knock down cells of type 2 Ip3 receptors, and we are in that process, label them in green, and see whether we can reproduce the results with 3D spheres, which would be quite nice. So now I'm just gonna, to conclude, I showed you that the type 2 Ip3 receptor is the minor subtype in GBM cells, and this is across either established cell lines or patient-derived glioma cells. And if we knock down the type 2 Ip3 receptors, and possibly through hypoxia, knockdown of type 2 Ip3 receptors leads to decreased cell-to-cell adhesion and increased invasion. As I mentioned, currently we're validating results in the patient-derived stem cells and in vitro invasion in brain organoids, and lots of questions remain open. We don't know yet how hypoxia downregulate Ip3R2. This would be really interesting to find out whether this downregulation is necessary for hypoxia to cause cells to detach. I'd also like to look at the mechanisms of the signals that pass through the Ip3 receptors to prevent cell detachment. How does hypoxia or loss of the type 2 induce cell detachment? As I mentioned, the data here is very preliminary, but we've done some mass spec on surface, biotin-related surface proteins, and we're sort of getting to some candidates that we're going to further check that might be responsible to maintain cell-to-cell adhesion, and the Ip3R2 might have some something to do with. And finally, so many of you know that this has been kind of my side project for several years now, but fortunately, this project has been fully funded by an MSc, a 3-year MSc grant from next October, which is joined with me, Colin, and Harry Bolstad. So this is where I'm going to be focusing for the next 3 years. And finally, I just wanted to thank, obviously, Colin, Laura, who is a PhD student in the lab, who's been working full-time on this project for the last 3 years. She's done a lot of the work with the GNS cells. She's carrying out the mass spec, and some of the data, the aggregation data that I've shown you comes from her. Anna Abrica, who was a postdoc who worked for 1.5 years in the lab. She did a little bit of the hypoxia work. And our collaborators, Harry Bolstad and Gemma, who's the postdoc who does both do the organoid work, and the funding bodies, and obviously you for your attention. Thank you.
Thank you very much, Anna. It's nice talk as usual. I don't know if Martin has something to say before we go to the questions.
No, please keep going, Raul. Thank you. Just keep going. Thanks a lot.
Can I ask you a question? Can I see can I stop presenting so I can see the people?
Sure you can.
Yeah, that's great. Thank you. Or maybe I just keep it in case I have to go back.
Yeah, just in case you need the slides to the question. So first question is from Martin. If you have any questions, please type it down in the chat box at your upper right corner. You still have time. So Martin asked that what's the difference? What difference did the Ip3R2 siRNA make to calcium signal in the C6 cells? Presumably, the Ip3R1 and Ip3R3 siRNAs have significant effects on calcium signals.
Mm-hmm. I have to say that this is something I started doing a long time ago and never sort of pursued. One of the issues is that we have some suspicion that the type 2 might be located in the nucleus, So the plan here is just to see whether the signals come from the nucleus and whether knocking down the type 2 wipes out signals from the nucleus. So the truth is a question that obviously we want to address, but I don't have the answer.
Okay, thank you. Mohammed Trebak, I'm sorry if I pronounce your name wrong, sorry in advance. So Mohammed asked, he has two questions now. So first one is, any correlation between Ip3R2 expression and invasion and also aggressiveness in public databases as ECGA, for example?
Yeah, so there's not a lot of data in terms of protein, but the mRNA levels seem to be down in more. So if you compare, for example, the grade 4 with less aggressive tumors, so that's data published in several databases. So yes, I didn't show that because it's not my data, but there's some correlation.
Thank you. So the second question of Mohammed is there any in vivo evidence of type 2 Ip3R role in glioblastoma progression in mice?
No, and that's obviously part of, so there's a step from going there to the mice, that's our idea of using the organoids, but yeah, eventually we will have to go to xenografts, but no, there's no data.
Thank you. So Vikas, I hope that's the pronunciation of the name, ask, does HIF bind to the Ip3R2 promoter and regulate its expression?
Yeah, that's something I don't know. We found HREs, like not the conventional, but some HREs in the promoter of the type 2. I'm trying to find out whether regulation is at the level of the mRNA. I don't have data to support that yet, so it might be just at the protein level, but this is obviously if we find that there's a downregulation at the mRNA level, we will obviously go that route of starting to look at HIF binding to the promoters, but no, I don't know.
Thank you. Jonathan has a comment more than a question. So he said, with the expression as low as it is, it doesn't seem to him that the expression is going to be a great indicator here.
The expression itself of type 2 Ip3 receptors.
I assume that that's it. So if we can actually answer that, will be great. I can go to another question in the meantime. So
yeah, I don't understand what the actual question is.
So it's not a question; it's more like a comment.
More of a comment. Okay.
So yeah, he said, like, with the expression as low as it is, it doesn't seem to him that the expression is going to be a great indicator here. I assume the Ip3R2 expression. So I don't know if you have any comment to add.
No, this has always been the interesting, a very interesting aspect, but it is true what he says. The levels. I mean, I wonder whether these cells are invasive already because they have such low levels of expression. And in the assays we do, reducing it further does increase invasion and thus decrease cell-to-cell adhesion. So there's some scope there to manipulate the levels. But I guess until we go to the in vivo data, we need to see whether this sort of is recapitulated there.
Yeah, Mohammed again asked, does the promoter of Ip3R2 has? It's the same question as before. So
yeah, I think yeah. So we haven't really. So we found through some programs some HRE. Like regions, but we haven't done anything to test, as I said, mainly because first I need to make sure that the down regulation at the protein level is matched by a down regulation of the mRNA level, which I don't have data to say.
And do you know if Ip3R1 or 3 has responsive elements?
Actually, no, I don't. I don't think we looked at R1 and R3.
Part of the question also. So Jonathan again asked, are there differences in the intracellular locations of the different IP3R subtypes in glioblastoma?
Well, so I didn't want to show this data because it is really preliminary, although I mentioned it. It's just one cell line where it seems that the all three show nice reticular patterns. The type 2 might be located in the nucleus, but I haven't done yet. So that's something that needs to be done, and across the cell lines to be sure. Because I mean, this antibody we have, I mean, I know that most people working with type 2 Ip3 receptor might agree that it's not an easy beast to work with. So we've got an antibody that's great for Western blot. It picks up the type 2 in the nucleus of one of the cell lines, but I am not convinced yet that it's specifically labeling the type 2 in the nucleus. So that is something we need to. It'd be really exciting if we could prove that actually is located in the nucleus.
Mm-hmm. Thank you. So Antonio Villalobo asked, is there any difference in the calcium toolkit used by cells undergoing 2D migration, like a wound healing assay, versus 3D migration, like through porous membranes and stuff like that?
So whether the differences in the calcium toolkit, I don't know that. I would assume so. Obviously, there are different mechanisms. So in the 3D assays, obviously, we have the sort of degradation of the ECM and so on, which is not present in the 2D. So I would have thought that, yes, there would be differences. That's why we also want to move to more sort of to assays where the whole thing is not just migration, but it's invasion, so degradation of extracellular matrix and so on.
Thank you. So Mohammed again, have you tried any of these invasion and spheroids experiments in glioblastoma lines established from patients?
Yes, so the last slide I showed, although as I said, it is preliminary because we don't have a full knockout, and they're very difficult to transfect these cells, so the siRNA wouldn't work. But yes, so there is indication that actually the phenotype on invasion is reproduced in these cells, which is quite exciting. And we're going to, once we get these cell lines, we're going to go to the organoids.
Thank you. John asked, any idea of the half-life of Ip3R2 protein in this cell type? Also, it is probably very long, he said, so meaning that the knockdown will take a long time to see at the protein level. Is this the case?
Well, so the knockdowns, we see it after 48 hours, gets better after 72 hours. I don't think we have like data to suggest what the time life is. I don't know. These are experiments we are planning to do, particularly if we see decreases in mRNA levels to see how far we need to go. But the knockdown is at 48 hours is quite good.
Okay, thank you. Anthony Morgan asked, does Ip3 sponge recapitulate the siRNA results? Is this calcium dependent or not?
We haven't tried that. It is a nice experiment to have in mind. As I said, I think I've been biased by seeing some of the type 2, although I'm not quite sure, in the nucleus. So I started looking at trying to measure the nuclear calcium signals to have something that wipes out the calcium signals and the nuclear independently, and then see. So I think I got stuck by looking at this, trying to go into details, and I probably should go and do these experiments. Now, I recall though that inhibiting PLC in the wound assays, although we all know that PLC inhibitors are quite dirty, did reduce wound healing, so suggesting that there might be a calcium signal.
Nick Yule asked, any possibility that the loss of R2 causes these effects because of a specific R2 binding partner, not necessarily directly the result of R2 calcium signal.
I couldn't agree more. Yes, so that's I would have done these experiments much earlier if it was with the type 1. But our problem, which I'm not sure if it's shared by everyone, but I guess so, it's just so difficult to work. So I have this experiment in mind where I want to do the poor mutant of type 2 Ip3 receptor and then see what happens. But with everything we've done to manipulate Ip3R2, the gene, has failed. We even tried to get companies to make plasmid for us, and that's why we're going through CRISPR. So no, that's definitely, I don't know, it could be that it's just, nothing to do with the calcium.
Sarah from Gil Lab asked, did you look at or quantify the changes in expression of beta-catenin?
No, we haven't. I did quantify changes in expression of encoding, and there's not massive changes, although the encoding story in GBM is quite controversial. So there's some studies suggesting that increase of encoding leads to more aggressiveness or invasion, and some others saying decrease. So that's kind of an area we're going. I think this mass spec is quite nice in that it's going to show us if it was to do with changes in the at least plasma memory expression of the adhesion molecules, we will see. So that's how we took that approach. We started looking. We didn't check beta-catenin, but the encoding didn't do anything. We knocked it down and we didn't see anything.
I don't know if this is a question for you or a comment for Anthony Morgan. This is from Martin, where he said nuclear targeted Hp3 sponge might be useful, right? For the previous question.
Yeah, is there? I'm not familiar with the. I mean, I remember when they first came out, and I haven't really looked. So, is there? I guess if you put a nuclear sequence, you would just. Yeah, that's a good suggestion, actually. I must say, I lack answers for this because, as I said, this has been some project that I really wanted to work on, and I was doing other things, but now. From now on, if the lockdown permits, I'll be trying to address all these questions.
Thank you very much. I think that's the last question. So, yeah. Thank you, Ana.
Thank you.